Thanks for coming to my talk. My name is Sriram. I'm a faculty here at PCA. Diving in. So I'm going to talk about a bunch of work that we've been thinking about on trying to do inference, scalable inference on large-scale genetic data sets. And I like this picture and also the title of this paper. It's quite old already. And basically what they show is how genome sequences are growing at the time. There's this super exponential growth of genome sequences, both in terms of number and in terms of the number of bases. And basically this paper was saying the total amount of genomic data is bigger than all the astronomical data, so probably you shouldn't talk about genomic data as being astronomical. We need a new lecture. All right, so what we are thinking about are these kinds of large genomic data sets, and as an example of that, there is the 1000 Genomes Project, which is thousands of individual genomes, tens of millions of SNPs, and then you have data sets like the UK Biobank, which is about 500,000 individuals from the UK, whole genome genotype, imputed data, so now it's again tens of millions of SNPs, and thousands of very diverse, rich phenotypes. And so the question is, we'd like to build scalable methods to analyze and visualize these genomic data sets. So, jumping right in, I don't need to introduce what SNP genotype data looks like, but this is like a typical genotype matrix. It's a matrix where every entry is a 0, 1, or 2. Every row is a SNP, every column is an individual. And I'd like to talk about some kind of common inference problems that we'd like to perform for data sets. So one problem I'll talk about is finding low-dimensional structure in genotype data. And the second one is, now we not only have genotype data, we also have traits, we have phenotypes on these individuals. And we'd like to learn some function that links genotype to phenotype. And Jahan's previous talk was also getting to this point. So let's talk about the first problem, which is finding low-dimensional structure. So this is an extremely important problem in population genetics, where this low-dimensional structure often has interesting information about the history of the population, about the ancestry, but it's also relevant to the second task, because often this low-dimensional structure tells us something about confounders that we want to control for when we are trying to learn these functions. So a very common task, a very common method for identifying low-dimensional structure is something called PCA, Principal Components Analysis. So basically, PCA takes this genotype matrix, and as an output, if you scale things correctly, you can get pictures that are informative of a person's ancestry. So this was a famous paper which looked at genotype data from individuals in Europe. And when you look at the first two principal components, so you take every individual and you plot them on this 2D plot, basically what you recapture is a map of Europe. And so this was a paper that had titled Genes Mirror Geography in Europe. And so there is interesting historical information in genotype data that this low-dimensional visualization can do. So now let's think about PCA and what are the mathematical problems and what are some of the limitations of this computation. So in PCA, we're given some vector. So xn here is a genotype vector of an individual n. It's a vector over m SNPs. And the goal of PCA is we want to write xn as a sum of k other vectors. So w1 to wk are these principal components. And we want to decompose each individual as a linear combination of these principal components. These numbers, zn1 to zk, are the coordinates of this individual along each of these principal components. So now we can massage this and write this in terms of a matrix W times Zn, which is the projection of this individual along the principal components. And this approximately equal to is because there is no guarantee that we can exactly write an individual like this. So there is an approximation that needs to be made. And so there's some loss of information. So let's make this more precise. So what's the problem that PCA is attempting to solve? So we want to identify the W and the Z's. So the W is the principal component matrix. So it's telling you what are these k low-dimensional representation. And Z is the projection of this individual along these principal components. So we want to identify both the W and the Z. And so the goal of principal components is to find a W and a Z so as to minimize this approximation error. And so precisely, you have your original data, your original genotype vector xn, and this is the approximation from PCA, and you want to minimize the error between the original data and the PCA reconstruction. So this problem by itself is not completely well defined, so we need to put some additional constraint. And the constraint is we want every column in W, which means every one of our principal components, to be orthogonal to each other. So that's this constraint. And so now if you actually try to solve this problem, the optimal solution, so the value of W and Z that minimizes the objective subject to this constraint, can be computed. And it turns out the way you compute it is you take the covariance matrix of your sample and you take the k largest eigenvectors, and that is the PCA solution. So the optimal W corresponds to the k largest eigenvectors of this sample covariance. So that's all great, and in fact, there are extremely good packages to actually compute PCA on any given data set. But what are some of the issues? A couple of issues. One is computational. So the way you actually go about computing these eigenvalues and eigenvectors for PCA is by running something called a singular value decomposition. And the question is, how expensive is this SVD? So essentially, the cost of computing the SVD scales either quadratically in the number of individuals n or in the number of SNPs n. So if you have extremely large data sets, that becomes infeasible. Now recently there's been a lot of randomized approximation algorithms for computing PCA. We'll talk about that. There's a second problem with PCA that's more from the fact that there isn't a very clear statistical interpretation of what PCA is trying to do. So PCA is solving a problem. The question is, what is the question, or what's the model under which PCA is solving this problem? And this can have quite a bit of impact. For example, when you have missing data, you have missing entries in your genotype matrix, how do you handle it? So there are many ways of handling it, and they make different kinds of assumptions, and PCA doesn't really tell you what is the right way to fill in missing data. It turns out the SNPs that we are dealing with, we have correlations amongst each other. It's often called LD. And for PCA to work, we need to decorrelate these SNPs. And there's a loss of information there. So it's unclear how do you actually deal with this correlation in a principled manner. So what we'd like to do is try to actually write down a statistical model which makes these assumptions clear and which still gives us a low-dimensional representation. So this is something that's been done previously. It's what's called probabilistic PCA. So it's a probabilistic version of PCA. It goes back to work almost 20 years back. And so what this model is it's a generative model. So it gives you a recipe for simulating data under this model. And then, of course, we are interested in the inverse problem, which is given data, we'd like to infer some hidden parameters. So what is this probabilistic recipe? So we're going to look at one individual. So for individual n, we're going to sample the Zn, which is this hidden variable, from some underlying Gaussian distribution. And now, given Zn and some additional parameters, we're going to actually compute our observed data xn. So xn comes as some linear transformation of Zn with some Gaussian noise. And I've chosen the notation here to match our previous notation because the W and the Z's here are exactly going to turn out to be the W's and the Z's we started off with when we defined non-probabilistic PCA. So we have a probabilistic model for one individual. We can now write down, given a data set of n individuals, we can write down this full probabilistic model.
When you say probabilistic, you mean a generative model? So the first randomization, or you toss Zn, and according to this Zn, you proceed deterministically?
Actually, there are two sources of randomness. So for every individual, you first draw Zn from this distribution. Then, having fixed Zn, or having picked Zn, and given these additional parameters, now you generate your xn according to this distribution. Okay? So it is centered around W times Zn with some noise. So the parameters of this model are W, Z W and sigma squared, and the hidden variable is Vn. And of course, we don't get to see any of these. What we get to see is Xn. A question at this stage? Okay. So the inference problem. So we have the probabilistic recipe. So inference problem is given X, we want to say something about hidden quantities. For example, we might want to figure out what is W and sigma squared. And again, given a probabilistic model, this is automatic in some sense. You write down the likelihood. How likely is your data X for some choice of your parameters? And then you search for this value that maximizes the likelihood. So in principle, that's straightforward. And what was shown in this paper, Tipping and Bishop, 1999, was that what you get by maximizing the likelihood is exactly the principal component. So that's nice, because it's a generalization, it has a probabilistic meaning, but at the end of the day, it gives you the same answer as doing regular PCA. So what have we gained by doing this probabilistic model? Well, if we go down this route and try to compute the maximum likelihood by actually doing eigen decomposition, we haven't gained anything, right? We're trying to get away from the computational cost of doing an eigen decomposition. But now that we have posed it as a probabilistic model, we have other ways of also computing maximum likelihood estimate. And one way to do this, when you have a model with this structure, so a model which has parameters W and sigma squared and a hidden variable Z, there's a very natural algorithm to perform inference. It's called the EM algorithm. So in any model which has these hidden variables, the EM algorithm gives you a very efficient or a straightforward way of doing inference. So what does the EM algorithm say? It says, let's decompose this problem into two parts. There are two unknown quantities. There is W and sigma squared that's unknown, Z that's unknown. So let's fix one and estimate the other, and use those estimates to re-estimate and keep iterating. So it's an iterative algorithm that moves between estimating either the Z's or the W's. So we can write down the EM algorithm, it's two lines. So we fix the W, estimate Z, take the estimate of Z and re-estimate W, and keep doing this till it converges. This is a special case where there is no sigma squared here because I'm assuming it. So this is just another way of computing maximum likelihood. Now the question is, have you gained anything by doing this? One thing that's worrying here is you're doing all of these matrix inverses. Always a worry when you're trying to scale up an algorithm. But it turns out, if we only care about computing the top few principal components, so let's say your data is 1 million by billion, but let's say all I want to compute is the top 10, 20, or 30 principal components. So K is, in that case, going to be 10, 20, or 30, N and M are of the million. And so this is the computational complexity of each of these. So what we have gained by doing this is each step of the EM algorithm is extremely efficient as long as you only want a small number of principal components. And for many of these data sets, that's often what we're after. Right? So when we're trying to compute the top 2, 3, 4 components, this is extremely efficient. Of course, there's also a question of how many iterations you need to run this. And it turns out the EM algorithm converges extremely quickly. So it's actually a very efficient algorithm. Turns out, can we do a little bit better if we know something more about the matrix? So basically what the E and the M step of this algorithm is doing is taking your genotype matrix, X, and multiplying it by some vector that keeps changing. In the E step, we take X and multiply it by some set of vectors, and in the M step, we take X and multiply it by another set.
What the size of W? Because W can be also huge, no?
So
is it unrelated to N and M?
So Z and W are going to be N by K and M by K respectively.
Okay.
So all of this works if K is small. So you're taking a big matrix and decomposing it into one matrix which is tall and thin, another matrix which is short. Okay, so it turns out, and I won't spend much time on this, because this X matrix is a genotype matrix, it's a matrix whose entries are all 0, 1, 2, you can actually make these multiplications even quicker. So there's a way of actually rewriting this matrix so that this multiplication, typically if it's an M by N matrix multiplication is M times N, you can actually make it go a little bit faster. And so if M or N are big, then that can be significant. But again, I can talk about that. Okay, so some benchmarking. So we did some simulations. This is simulations of different populations that have diverged over some period of time. So FST is telling us how diverged these populations are. So as you go from bottom to top, the populations are getting closer. So it's harder to estimate principal components. And so the goal is, can we estimate 5 or 10 principal components on this data? And this metric is telling you how close is what we get out of this algorithm compared to running an SVD. So closer it is to 1, the closer are the 2 sets of principal.
So if you do enough iterations, are you going to converge to the right value, or do you still have this? Or is it
This one?
Multiple boxes.
Yeah, so you do need to do enough iterations to be able to converge.
So the error you're making here, is it convergence, or is it that you don't find the right optimum?
Yeah, he probably meant not iterations, rather runnings, in order not to be kept. I mean, single maxima or multiple maxima.
Oh, so actually, at least, well, there's no guarantee that you don't converge to local optima. But what we have seen is, even with random restarts, you pretty much converge. The question is how close, how many iterations you need to converge.
Converge to the same point all the time.
Yeah, similar point. I mean, they're very, very similar. But, you know, I guess, yeah, in general, even for relatively close FSTs, so these are relatively close human populations, we get quite accurate principal component estimates. So then there's the notion of computational cost. So here we've varied the number of individuals, and we're asking what's the computational time. So this is if you run different kinds of algorithms, and this is if you do a full SVD. So what you see here is nonlinear scaling, which is what you expect. And then there are a bunch of different algorithms which are all iterative. So we have EM, then there are these other algorithms which are based on these randomized projections. And they're all linearly scaling, so they're all quite good. Turns out the EM is still a little bit faster than these other algorithms, depending on how you want to look at it. So then we applied this to UK Biobank. So this is an example run. So this is about 500,000 individuals in the UK Biobank, 150,000 SNPs. So this is not all the SNPs. This is LD prune, so SNPs that are decorrelated. And so you can compute top 5 PCs within about an hour. And the principal components look reasonable. So the first principal component is separating African, non-African ancestries. The second principal component is separating out different non-African ancestries. All right. So the nice thing about UK Biobank is you can actually compare the principal component estimates with estimates of geographic location. So individuals come with sampling location. And so now for every individual, you have the principal component estimate and you have their sampling location. And so you can compare the two to see what these principal components are actually picking up. So here, for example, are the first principal component value for every individual. And basically, again, it's a little bit saturated, you'll see that there is kind of a north-south gradient that this principal component is capturing, although there's also some Wales effect.
For this image, did you remove like non-British people from this plot?
Yeah, so this is all the white British individuals in the UK. Yeah, so it's like 300,000 odd.
And it was the same first PC that you showed us before, or did you rerun the PC on these you have individuals?
Yeah, so that was on the full data set. Now this is on the white British.
You rerun the PC.
Yeah, so this is rerunning on the white British individuals. Yeah, otherwise you wouldn't pick up these kinds of differences if you ran it on all the individuals. Other interesting things you can potentially do with these kinds of fine-scale population structure is it's interesting to ask what are SNPs that are not well explained by the principal components that you have detected. So every SNP has some kind of projection on each of the principal components, and you can ask, are there SNPs that are not well explained? Typically, most of the SNPs are artifacts. So there's like genotyping errors, there are regions of long-range LD, so we have to do a round of tuning and QCing. And at the end of the day, we still find some SNPs which we think are not artifacts and are not explained by the principal components. So we have a statistic which is measuring kind of this residual of every SNP relative to each principal component, so PC1 through PC5, and we can plot the QQ plot of the statistic, and it's relatively well calibrated, and then there are these tail of SNPs that are not well explained. So now we can see what are these classes of SNPs that are not well explained by each of the PCs, so PC1 through 5, and there's also a combined statistic. And you'll see that there are SNPs that are known candidates for previous signals of positive selection. For example, there is the lactase gene, that's known to be a target of very strong recent positive selection. There is the DLR gene, which is an immunity-related gene, HERB2, which is eye color related. And there are a bunch of others which have been less well characterized.
Sorry, so I guess I'm not clear on what the different colors here are. So that means it's a SNP that isn't explained by PC1, but could it be explained by PC2, or is it not explained by all of these at the same time?
Right. So we do this for every PC separately, and then there's a combined statistic. So yes, it's possible that a PC is well explained, a SNP is well explained by PC1, but not by PC2 and so on. So in other words, going back to a geographic example, if you look at PC1 as a north-south gradient, then a SNP that's not well explained might have, say, very uniform distribution or a much more extreme distribution north to south in UK. So this is potentially interesting because this is telling us within the UK, within the population structure that you see here, what are SNPs that are potential targets of positive selection. And this is these kinds of statistics have been recently used, and so there's a paper from Marcus Price's group that looks at these kinds of statistics. We talked about a probabilistic model as being good for handling missing data. So this is just a simulation to show the value of having a probabilistic model that handles it. And basically all we are showing is, as a function of FST, how accurate are your principal components. You do some kind of imputation, fill in your missing SNPs and then do principal components, versus let the probabilistic model automatically impute. And generally there is a small improvement in the accuracy when you allow this probabilistic assignment of missing. Okay, so just to conclude this part, so we still haven't solved some of the major problems in this framework. So one question is, how do we actually handle correlations amongst SNPs? Clearly, we are throwing out a lot of information. You have large data sets, you have rare variants that are being thrown out by this kind of procedure. So how do you actually model this kind of correlation in a probabilistic framework? And one thing that is always a bit of a problem is the probabilistic PCA is not an exactly good fit for SNP genotype data. SNP genotype is discrete. Probabilistic PCA is making continuous observations, so there is a mismatch. So ideally, we'd like to be able to replace the normal observation model in probabilistic PCA with a discrete model, which outputs zeros, ones, and twos. Turns out, in that case, you don't have a lot of the nice exact inference that we talked about. So there are issues to be solved when you actually try to model the data exactly. All right, so with that, I'll move to the second part, but any questions before?
So, I mean, general questions like last week we heard in the single cell context for the visualization and low dimensions, people are moving away from PCA and we have all these other techniques. Are people exploring these in the context of the genetic data and not by
Someone has a paper, preprint on that. Yeah. So there is, I guess, people haven't explored it as much, but I guess there is definitely a lot of room there.
You work well.
Yeah.
Yeah, so I was wondering, when you say that there are some SNPs that aren't explained well by principal components 1 through 5. So if you were to do this and you look at only principal components 1 through 4, then are the majority of the leftover SNPs explained by PC5? So that is to say, are the residual SNPs, would they actually be explained by the next principal
Right.
component?
Yeah. It's not the case that this is just explained by lower PCs. And the reason for this is these components are orthonormal to each other. So we are looking at, so these are explaining variation that's orthogonal to the variation along each of the principal components. And so it is not the case that as you move down, you suddenly find more of these steps being explained.
Okay. Thanks.
What is the test that you were doing in that there's like a log p-value on the y-axis?
Right.
The SNPs, what's that test?
So basically what it's doing is you have the original value and you have the reconstruction at that SNP from the principal components.
But just from one principal component,
Right.
if you have blue or something.
Right, but then you project it onto that principal component. So you take the residual and you project it onto that principal component and see how much is explained along that principal component.
And what's the p-value?
And so if it is well explained, there should be like the sum of squares of those residuals is chi-squared 1 degree of freedom. And so then you have a p-value associated with those results.
So you said sigma square goes to 0, so basically you're not assuming that sigma square goes to 0, right? Otherwise, with the okay principal components, you should get an exact reconstruction. So you need the sigma square though somewhere in your chi-square statistic.
Yeah, so sigma squared is soaking up all of the residual variation. So you have the sum of all of the principal components, the average principal components,
Sure, yeah.
from k plus 1 onwards.
But how do you estimate sigma square? You just estimate it empirically from your residual?
Yeah, you can estimate it from the residuals.
So you need some kind of robust estimate if you want to assume that those outliers.
That's right. So if you have a large number of outliers, you would need a robust estimator. Yeah. So given our QQ plot is fairly close to diagonal, we've just used a simple average. All right. So now let me talk about the second part. So now let's bring phenotypes into the picture. So the question we are going to be interested in is trying to associate phenotype with genotype. So a basic question here is, for example, how much of a phenotype is determined by genetic factors? And one way of making that more precise is we'd like to figure out what is the best predictor, a linear predictor of phenotype from genotype. And that quantity is called heritability. So this is an area of active research, and heritability is one number, but there is a lot of work in trying to go beyond just quantifying heritability and understanding it in different contexts. So for example, people have looked at the heritability of a phenotype across different chromosomes. So for example, heritability of schizophrenia as a function of chromosome length. You see this relationship, which has been interpreted as a sign of polygenic architecture. You can look at heritability in finer regions of the genome. So this was a paper that looked at 1 megabase regions and quantified heritability of, in this case, schizophrenia in 1 megabase regions of the genome. You can look at pairs of phenotypes and quantify what is heritability that's shared. So it's a concept called co-heritability or genetic correlation. And then you can ask, if I divide up the genome into different categories, coding versus conserved regions, what is the proportion of heritability or an enrichment of heritability that comes from different regions? Okay. So clearly, this concept has been used in lots of different studies in trying to understand genetic architecture. So what's the underlying assumption? So you have a set of individuals. So you have with phenotype, you have a genotype matrix, and we are assuming a linear model. So each SNP has an effect size given by beta plus environmental noise. And here we're going to assume it's the phenotype is Gaussian, so the noise comes from this Gaussian distribution. Turns out typically the number of SNPs is going to be much larger than the number of individuals, so you can't estimate the parameters in this model. So we need to make additional assumptions. So typical assumptions include putting a prior on these betas. So a very common prior is you assume that every entry of beta, each of the SNPs, comes from this distribution, zero mean Gaussian with some beta. So with these assumptions, so assumptions on the SNP effect size and the environment, now heritability can be defined as the ratio of these two. So you can think of sigma g squared as the effect size associated with a SNP, and sigma e squared is the variance associated with the environment. And for those of you who worked in other contexts, in non-genetic contexts, you can think of heritability as a measure of signal to noise. So signal is the proportion of variance that's explained by the features, and noise is everything that's not explained. So this model is something called a linear mixed model. And so this is the formulation. So you have your phenotype coming from x beta plus noise, and beta has some prior on it, and the noise has a Gaussian distribution. So now the goal is we'd like to be able to estimate heritability, and then we'll talk about other generalizations. So to estimate heritability involves estimating these two variance parameters. Again, we are in a probabilistic model, so that's nice. So we can write down the log likelihood. So we can write down log likelihood with these two parameters. So that's the probability of your phenotype given genotype and the parameters.
The likelihood is over the two sigmas, which actually this is your heritability. These two sigmas give you, sigma square, give you the heritability, correct?
Right. Yeah.
So this is the likelihood of the heritability. Is that correct?
Well, the heritability can be defined.
Exactly.
Yeah, if you parameterize it, you can define it.
Okay. So this is, so the likelihood is over these two parameters.
That's right. And now we take this likelihood and write it in terms of two quantities. So the probability of the phenotype given effect sizes, genotype, and environment variance, and the probability of the effect sizes, and we integrate out the effect sizes. So all of this problem would be easy if you knew which SNPs are actually affecting the phenotype. We don't. And so that is why we need to integrate out over the effect sizes. So again, we can ask how easy is it to evaluate and maximize this objective function. Turns out it's quite computationally expensive because when you actually evaluate this likelihood, you have to, this is a Gaussian distribution over n individuals. And so you have to actually compute and invert an n by n covariance matrix. So because of that matrix inversion and there's a determinant computation, every evaluation of this scales as n cubed, where n is the number of individuals. And of course, there has been lots of work in trying to speed it up and make it more efficient. But in general, you still have to perform this in computation. So now I'll talk about an alternate way of estimating this, and that has some trade-offs, and the upside is it's going to be more efficient. So an alternate way of trying to estimate parameters in a statistical model is, instead of trying to maximize this likelihood, you try to match up the different moments of this model. So moments of a model would be the mean, the covariance, and so on. So for example, in this model, the first moment, the mean of y, the way you set it up. So if you look at the original model, the mean of y is 0, so there's no information there. So let's look at the second moment, so the covariance. So we can write down the covariance of y, and if we do some algebra, we can relate the parameters we get. So sigma g appears here, sigma e appears here. Okay. So in other words, if I can estimate the covariance y, then I can try to fix values of sigma g and sigma e so that they best match the covariance. And the key quantity here is the matrix K. It's an n by n matrix that is formed from the set of genotypes. It's called the genetic relationship matrix. Basically, it says for every pair of individuals, how closely are they related. So now the goal is, if I can put an estimator of the covariance matrix, and then I try to choose values of sigma g and sigma e that match up the estimator of the covariance. So in this setting, there's actually only one estimator. We have one sample from y. So the estimator of the covariance of y is just this quantity. So we just have one sample, so this is the trivial estimator of the covariance. So the goal now is, let's pick values for these two, so that this quantity matches this quantity. So now you can actually solve this problem. It turns out this problem has an analytical solution. So it's called a method of moments estimator. And basically solving it involves solving a set of two equations in two variables. So this is a linear system, two variables and two equations, so we can solve it analytically. So method of moments is something that you can solve analytically, but there's still one catch. You still have to compute the GRM. Remember, the GRM is an n by n matrix, and it's formed by taking the genotype matrix for every individual and computing enough products. So that is still expensive. It's an n square n operation. So we still haven't come up with a solution that scales. Now, the main observation is really, we don't need to compute the GRM. We only need to compute traces of the GRM and the square of the GRM. We only need these two numbers. And so we're going to approximate them. So this leads to the randomized method of moments estimator. So the basic idea, this is a linear algebra problem, is if you have the trace, if you want to approximate the trace of the matrix, there is what's called a randomized estimate of the trace. So the trace of k squared can be approximated by taking z, which is a random vector, which would be mean zero, covariance identity, and computing z transpose k squared z. You do that b times the new trace. So that's an unbiased estimator of the trace of this matrix. Turns out the trace of k is known to us because the genotype is standardized. So all that needs to be done is we need to compute this, and with a little bit of algebra, it turns out this can be computed without ever forming the K matrix. So all we need to do is multiply the genotype matrix with some random vectors b times. And if your b is small, if you need a small number of random vectors, then this has saved you some computation. So first question is, how many random vectors do we need? So this was some simulation on a data set of about 5,000 individuals, trying to estimate heritability as a function of the number of random vectors. And it turns out we don't gain much by increasing the number. So if you can go as small as 10, we rarely see any gains in going beyond 100. So typically, 10 and 100 are much smaller than 500,000, so that's a bit of a gain. What about the accuracy? So we said there are these two ways of estimation, maximum likelihood. We gave up on that, we did method of moments. So there's no free lunch. It turns out method of moments in general is less accurate than maximum likelihood. And that is something we see. So here is a simulation for different values of the true heritability. What is the estimate? We compared GCTA, which is a maximum likelihood or REML estimator, with the method of moments. And you see your standard errors are a little bit bigger. So roughly we find that it's maybe a 10 to 20% increase in the standard errors when we use method of moments.
But your true heritability is dependent on the two sigmas. So how they affect, so how your true heritability, or let's say the entire right-hand figure, how it is defined in terms of the two sigmas, not in terms of the true heritability.
Right, so in all of these simulations, we've chosen the denominator to be 1.
Okay.
So, and then we play with the numerator. All right, so, and then, of course, the question is it faster? And for these kinds of small number of random vectors, it is much faster than current methods. So we compare it to both REML, GCTA, and so on. And again, it can run extremely quickly on datasets like the UK Biobank. All right, so it was all about getting point estimates. Of course, for this to be useful, you also want to compute standard errors. Right? You want to figure out what's the error bar around these estimates. There are many ways of doing this. We use block jackknife. So basically, what you do is you take your genome, you drop blocks of the genome, and re-estimate, and use those estimators to build up an estimate of standard error of your original method of moments. One reason why this is a good thing to do is this doesn't actually add any additional computational cost. So you can do this without actually explicitly running this 100 times for 100 different blocks. Another thing about this algorithm, which is nice for these large data sets, is actually what we call a streaming algorithm. So if you noticed, once you compute this trace of k and trace of k squared, that's all the information we need. So you can just read your genotypes a few SNPs at a time and build up your estimates of these traces. And so, in fact, the memory requirements are fairly modest. So you can run these on large data sets without running into memory issues. All right, so in the next couple of slides, I'll just talk about a few extensions of this. The basic idea is the same. It turns out it can be extended in a bunch of different settings. So as I said, you're interested in not just heritability, but how it varies along the genome in different kinds of functional annotations, for example. So for example, we might want to have a different variance parameter for those SNPs that lie within a gene versus those that are outside. So that is a problem of partitioning heritability. So it's a multiple variance component problem. So now instead of just sigma g, sigma e, you have maybe k different sigmas corresponding to which component your SNP lies in. Everything you've said carries over to the setting. Now instead of two linear equations and two unknowns, you have k plus 1 equations with k plus 1 unknowns, and you can solve it efficiently. I go quickly over this. So it turns out there's a lot of debate in human genetics community about what is the right way to estimate SNP heritability. And one of the things that's coming out of that debate is the fact that you want to fit models which allow for different variance parameters depending on the allele frequency and the LD patterns on the SNP. So one thing that is recommended is to actually have variance components that vary depending on these annotations. And so here, this was a bunch of simulations with different parameters. I won't get into the details, where we actually controlled how these SNP effects vary. And the methods that we compare to are all slightly different. They're summary statistic methods. They're not individual methods because they couldn't be run on UK Biobank. And the basic takeaway is it matters that you actually partition your variance according to LD and allele frequency estimates. And so this is like showing the bias of the estimators depending on whether or not you can partition it. This is small scale data where you can compare method of moments to maximum likelihood. So method of moments is blue, maximum likelihood is red. And again, you see that there is an increase in the standard errors for these kinds of smaller data sets. And of course, this is just the runtime. It is very scalable. It can also fit hundreds of components. So that's something that's quite useful. So then we ran it on actual traits in the UK Biobank and compared it to current estimates from other methods. And again, I won't spend too much because this is, the details matter here. But in general, we find that our estimates are slightly lower than estimates from other methods. And of course, I should also point to a paper that's coming out from Bogdan Sanovich's group, where they have an estimator which is robust to a lot of these assumptions. But of course, more interesting is what happens to these heritability as you partition them along the genome. Are there interesting things we can show about the genetic architecture? So here we partitioned it based on common SNPs and rare SNPs. And within each SNP, we partitioned it based on what's called the LD score, which is the sum of these of the LD of the SNP to all of its neighbors. So these are SNPs which have high LD score versus low LD score. And so you see this pattern where SNPs which have low LD scores are enriched for heritability. And this is true both in common and the rare allele frequency spectrum. And of course, this has connections to selection. And the question is, what does it mean in terms of understanding traits in the context of selection? This can also be done with multiple traits. I'll skip this in the interest of time. And then there's also extensions to a nonlinear fixed model. So I think that's actually quite interesting. And the only one reason to actually talk about this is these ideas came out of a bunch of discussions at a couple of meetings. So there was the meeting at Partitioning organized last year, where a lot of these ideas came about, and at last CGSI and the CGSI, a lot of these details have been worked out. And the basic idea is now you want to expand your model. You don't want to just have X times beta, you want to have, say, all pairwise combinations of SNPs. Or you might have some environment that you measure, and you want to have all pairwise combinations of SNPs and environment. So clearly, you can fit all of this. The question is, how do you avoid the blow-up? So if you have all pairwise combinations, you have m squared features, or m times L features. And so then even linear in that setting is not good enough. But it turns out there is additional structure in this problem that allows us to make these estimates without actually explicitly quantities. So you can actually compute these kinds of quantities in time which is linear in the original number of SNPs and the number of individuals. All right. Still, there are lots of open problems. We'd like to better understand these randomized estimators, try to understand how robust they are. They're efficient. But of course, their efficiency depends on these kinds of specific linear mixed models we've assumed. What about nonlinear kernels? Can we actually do other interesting models here? And of course, you might not care about genetic architecture. You might not care about these variance components. But one reason you might care about some of this is important in prediction. So you can think of this as step one in doing trait prediction. So can this be used for doing prediction on large data sets, or in doing association testing? Again, this is like a sub-problem that you end up solving. So can this be used for improved association testing? So this is just summarizing a bunch of what I said, and I'd like to thank the students. So the PCA work was done by a very talented undergraduate, Aman, and Alec, who's a bioinformatics PhD student. The work on randomized method moments was done by Ariel and Lee. And I'd like to thank all the funding and mathematicians.